Good day everyone, we are group number 5 and today we are going to report on activity number 2, the thermodynamics of protein unfolding. In this activity, we used calorimetric measurements to determine the free energy of protein unfolding. To start, let us first recall the principles of differential scanning calorimetry. Differential scanning calorimetry is an experimental method that directly measures the difference in heat energy uptake taking place in a sample relative to a reference during a regulated temperature change. This method is carried out in a differential scanning calorimeter, which involves the introduction of heat energy into a sample cell and the reference cell simultaneously, while identically increasing the temperature of both cells over time. The underlying principle here is that, due to the difference in the composition of the sample and the reference, different amount of energy will be required to raise the temperature of the cells. Therefore, the excess amount of energy required to compensate for the temperature difference between the cells is measured and directly correlated to specific thermodynamic properties of the sample. DSC is a technique used to characterize the stability of a protein or other biomolecules directly in its folded form. It does this by measuring the heat change associated with the molecule's thermal denaturation when heated at a constant rate. A biomolecule in solution is in equilibrium between its native and denatured conformations. The higher the thermal transition midpoint or TM, the more stable the molecule. DSC measures the enthalpy and the change in heat capacity of unfolding that results from heat-induced denaturation. It can then elucidate the factors that contribute to the folding and stability of native biomolecules. These include hydrophobic interactions, hydrogen bonding, conformational entropy in the physical environment. Macromolecules and macromolecular assemblies such as proteins, nucleic acids, and lipids can form well-defined structures that undergo thermally induced conformational changes. These structural rearrangements result in the absorption of heat caused by the redistribution of non-covalent bonds. Differential scanning calorimeters measure this heat uptake. The enthalpy of protein unfolding is the area under the concentration normalized DSC peak. In certain cases, thermodynamic models can be fitted to the data to obtain the Gibbs free energy, the calorimetric enthalpy, the entropy, and the change in the heat capacity associated with the transition. These thermodynamic parameters can be calculated using the following formulas. Here is a general methodology in performing a DSC measurement. First, the reference chamber is filled with buffer and the sample chamber is filled with sample. Then, the experiment parameters are set up, the chambers are then heated at a constant scan rate, and then the data are generated and analyzed. The objectives of this activity is to discuss principles of differential scanning calorimetry, calculate thermodynamic parameters of protein folding, and to interpret DSC data on bovine serum albumin unfolding and relate the findings with protein structure and function. Now let's proceed to activity 2.1, the analysis of thermodynamic parameters for various proteins. Here is a table for the given thermodynamic parameters for the unfolding of some proteins. For the first guide question, observe the per residue thermodynamic values in the table. What can you say about the values? From the table, we can observe the high values of delta H naught per residue. This is because protein folding is stabilized via hydrophobic interactions between residues, and therefore protein unfolding is an endothermic process. Since the process is non-spontaneous, protein unfolding must have positive values for its free energy at 25 degrees Celsius and relatively lower values for its entropy at 25 degrees Celsius compared to its enthalpy at 25 degrees Celsius. Meanwhile, there seems to be no direct correlation between molecular weight and entropy at 110 degrees Celsius. This observation means that the unfolded states of these proteins are somewhat similar to each other, and by extension, so do their folded states. As for Cp, a positive value correlates to the amount of nonpolar groups exposed to water upon unfolding. The more positive Cp is, the more nonpolar groups are present in the protein. It can then be inferred that the hydrophobicity of myoglobin is the highest among the proteins in Table 1, with ribonuclease being the least. As a consequence, proteins with higher Cp tend to become more globular to minimize surface area. For the next question, graph the delta H naught and negative T delta S naught against the temperature of your assigned protein. Comment on the behavior of the two curves. What is the significance of the intersection of the two curves? So the assigned protein to our group is the hen lysozyme. From the given thermodynamic parameters in Table 1, we determined the delta H naught at 25 degrees Celsius, delta S naught at 110 degrees Celsius, and delta Cp of the protein. Using these formulas, we determined the delta H naught unfolding, delta S naught unfolding, and delta G naught unfolding at different temperatures, and the calculated thermodynamic parameters are summarized in Table 2. By graphing the delta H naught and the negative T delta S naught against the temperature of the hen lysozyme, we can see from the graph that the delta H naught increases with increasing temperature, while the negative T delta S naught decreases with increasing temperature. As for the intersection of the curves, this is the temperature where delta H naught is equal to negative T delta S naught. At this point, cold denaturation starts to occur. Cold denaturation is when the protein starts to denature due to the cold temperature. Beyond this point, delta G naught unfolding and delta H naught unfolding begin to become negative, and unfolding becomes thermodynamically favorable. This is because when temperature is decreased, interactions between water molecules and nonpolar amino acids become favorable. This is consistent with the delta G naught versus temperature graph on the next question. Shown in the slide is a graph of delta G naught against temperature. So from the graph, we can see that the peak is the temperature of the maximum stability of protein, and beyond this point to the left, the stability of protein decreases when T is decreased. Thus, we can conclude that denaturation starts to occur. For the last question, we determine the Tm of the protein. At delta G naught equals 0, the temperature is equal to the Tm. Hence, by letting the delta G naught of the equation become 0, we can calculate for the Tm. In this case, the Tm is 90.42 degrees Celsius.
Let's now head to the second activity, with the title Calorhythmic Measurements on Bovine Serum Albumin. But what is bovine serum albumin? Bovine serum albumin, or BSA, as shown in the image, is a protein dissolved in blood plasma and is the most abundant blood protein in mammals. It functions as a cell nutrient and is used to stabilize enzymes during restriction digest. It also acts as a transporter that binds and carries small molecules such as fatty acids, sending them between cells and tissues. In this experiment, we will determine the thermodynamic parameters of the unfolding BSA when it is associated or dissociated with fatty acid using a DSC profile, then correlate the values obtained with the stability and function of BSA in organisms. We labeled the BSA with fatty acid with BSG, while BSFG for BSA without fatty acids. We then extrapolated the thermodynamic parameters based on the graph and calculated their corresponding values at the physiological temperature, or at 37 degrees Celsius. At melting temperature, the folding and unfolding proteins are at equilibrium, meaning their equilibrium constant will be 1. Upon substitution to the Gibbs free energy equation, which is delta G of unfolding equals negative RT ln K, we will obtain a value of 0. Thus, Tm can be located at the peak of the curve where delta G is 0. Now, to get delta Cp of unfolding or change in heat capacity, we can get the difference of Cp between the starting point and the end of the curve where the sample is concentrated with folded and unfolded proteins respectively. Then, for delta H of unfolding or change in enthalpy, we can integrate the Cp over the curve or simply get the area under the curve to determine the total amount of heat taken as the folded protein is converted into unfolded one. After extrapolating delta H, we can now calculate for delta S or unfolding or change of entropy by using the Gibbs free energy equation at equilibrium, which is delta G equals delta H minus T delta S. Substituting 0 to delta G and rearranging, we can get the value for delta S unfolding as delta H divided by melting temperature. To make the extrapolation easier with relatively higher accuracy, we utilize an online tool named Sketch and Calc. These are the figures obtained from Sketch and Calc for the BSG and BSFG. We obtain the values for the area under the curve, Tm and delta Cp with a scale 1 unit is to 1 degree Celsius is to 0.08 millijoules per mole Celsius. Using the equations and methods discussed earlier, we now extrapolated and calculated the values for the melting temperature, change in heat capacity, change in enthalpy, and change in entropy of unfolding. For BSG, shown on the right are the computations we made with respect to the scales used in plotting. We obtained a Tm of 71.67 degrees Celsius, 1.08 millijoules per mole degree Celsius for delta Cp, and 54.43 millijoules per mole and 0.76 millijoules per mole degree Celsius for delta H and delta S respectively. Meanwhile, for BSFG, we obtained the following values: Tm of 56.27 degrees Celsius, 1.54 millijoules per mole degree Celsius, 30.1 millijoules per mole, and 0.54 millijoules per mole degree Celsius for delta Cp, delta H, and delta S respectively. Finally, for the parameters change in enthalpy, entropy, and free energy unfolding at 37 degrees Celsius, we calculated them using the equations below. Note that we use Kelvin in the computations. We then obtained the following values for BSG: 54.29 millijoules per mole, 2.35 microjoules per mole Kelvin, and 53.56 millijoules per mole for delta H, delta S, and delta G unfolding accordingly. Meanwhile, for BSFG, the values were 29.99 millijoules per mole, 1.61 microjoules per mole Kelvin, and 29.49 millijoules per mole for delta H, delta S, and delta G unfolding respectively.
Before we discuss the values calculated just now, let us first discuss why Tm is a prominent parameter in DSC more so than delta H or delta G. Melting temperature, or Tm, is defined as the temperature at which the fractions of folded and unfolded states of the protein are equal. As demonstrated before, Tm is the easiest and most accurate parameter that can be measured from a DSC curve. Tm is more often used as a measure of protein stability as it is not concentration dependent. Moreover, calculations for delta H, delta S, and delta G are dependent on Tm and baseline construction. Tm can also be a distinguishing feature between unknown protein samples, buffers, and substrate binding affinity. Now that we know the implications of Tm, let's look at our data again. An increasing Tm and delta H of unfolding means that the BSA fatty acid complex is more stable than BSA alone. This makes BSA an effective fatty acid carrier. In fact, it can carry up to 7 fatty acid molecules at a time and transport it to adipose tissues or to distant parts of the body when needed. In comparison to the size of BSA, which is about 583 residues long, the contribution of the bonds of the fatty acids and the coordination of the active site to each of the 7 molecules should be negligible. Therefore, we can conclude that the stability of the BSA fatty acid complex is mostly due to the rearrangement of the protein's residues. A similar observation is made when we calculate for delta G at 37 degrees Celsius. With the more positive free energy of unfolding, BSG is more stable than BSFG inside the organism. With that, we're almost done with the presentation. There's only one part left. Along with the discussion, we know that the differential scanning calorimetry is a reproducible technique for identifying the thermodynamic properties of protein unfolding. Specifically, we were able to determine that melting temperature is a reliable measure of protein stability that is independent of concentration, unlike delta H. We also know that calculating for delta G over a wide range of temperatures shows the lower limit of protein stability before cold denaturation takes place. And lastly, we know that substrate binding noticeably changes the protein's DSC curve. This is helpful for determining the effect and favorability of other substrates, as well as inhibitors and activators. That is all. Thank you for listening, and here are our references.